Hi everyone, I'm Ramin, a third-year PhD student at UBC, and today I'll be presenting our work Ccrl, or Contrastive Cell Representation Learning at Eccv. This work addresses one of the main challenges in cell representation learning in the context of histopathology images using self-supervised learning. Recently, the digital processing of whole slide images, or WSIs, has attracted a lot of attention from machine learning and computer vision communities. And the reason is that this domain has a lot of new challenges. For example, one of these main challenges is that these whole slide images can reach to around 100,000 by 100,000 pixels in size. So this size is going to cause a lot of problems at different stages of common machine learning pipeline, starting from storing the images. So to store the whole slide images, we usually use a pyramidal shape similar to what you can see in the left figure. This means, starting from a low resolution image, we can gradually zoom into different scales of magnification and take a look at the tissue structure and also cells. But storing the images is not the only problem that comes with this size, but it also introduces problems in the processing of whole slide images. So the conventional paradigm for processing of whole slide images includes using multiple instance learning, or MIL approaches. More specifically, these techniques divide the whole slide image into smaller patches and then pass these patches through a convolutional neural network to get the corresponding representation of each patch. Finally, using an aggregation method, which can be a simple pooling operation, like mean pooling, attention pooling, or even a variational pooling, they represent the whole slide image with only one single vector. And finally, we can use this single representation for downstream tasks like cancer type classification, survival prediction, or even mutation prediction. Even though this model works pretty well, there are actually a couple of questions that need to be answered in this domain. The first and the most important question is the concept of biological entities. As we all know, cells are the main components of the tissue that build its characteristic. But as we saw, MIL approaches mainly focus on patches, which means they use the pattern appearing in the population of the cells. This also means they would lose information about the individual cells, including cell locations, their types, and more importantly, the mutual interactions that exist between the cells. However, many studies have shown that this type of information can actually be quite important and critical for tasks, especially tasks like survival prediction, where we need detailed information about the context of the tissue. So this tells us that we can kind of change our perspective from patch processing to cell processing. But obviously doing so requires coming up with an idea or a model to find the identity of individual cells. So the first thing that comes to someone's mind can be using supervised cell classification approaches like HoverNet. But obviously using these approaches is not practical and also scalable, because to train a supervised model, we need to have a large, fully annotated data set in which all the cells are manually annotated. In a 30-second video, we can see more than hundreds of cells in one whole slide image. And this is around 5% of one single whole slide image. So if we wanted to do this for multiple whole slide images, it's going to be very, very time consuming and also expensive. So clearly, this is not the way to go. But even if we put all of our effort into collecting such a data set, it's going to be dependent on a specific cancer type and also the setting that we use for dataset collection. So if we wanted to apply this model on a new cancer type, we would have to go through the dataset collection and annotation process again. And obviously this is not feasible. So to address this issue, we came up with the idea of using self-supervised learning in the context of cell representation learning. More specifically, first, we're going to use an unsupervised approach, like color thresholding, to locate all the cells and then take a small crop around each cell, similar to what you can see in the figure. Then, for each cell, we're going to apply two sets of augmentation. one whole view augmentation and the other one partial view. Then we're going to pass the whole view through an online encoder and then a prediction head to get its representation. Also for the partial view, we're going to pass it through a momentum encoder. Finally, using a contrastive loss, the formula of which you can see in the figure, we're going to bring the embeddings of these two images close to each other as much as possible. There are three important parts in this design. The first part is the whole view augmentation. This part helps the model to build a local to global relationship between the representations of the cells and make it work better. The second part is the prediction head, which gives the online encoder more flexibility. And finally, the last part is the momentum encoder, which technically is an averaging of the weights from the online encoder. In other words, it's going to be an ensembling of the online encoder, and because of this reason, it's going to be more robust and more accurate in representing the cells. So we're going to use this momentum encoder at inference time instead of online encoder. So we tested our model in the task of cell clustering on 3 different data sets from 3 different cancer types. We also used adjusted mutual information, adjusted rand index, and purity for doing this comparison. As you can see, our model, Ccrl, can outperform all of the existing baselines across 3 different data sets. And these baselines also include 2 state-of-the-art deep learning models named autoencoder and IFOGAN. Also, we conducted an ablation study on our experiments and saw that all the 3 parts that we talked about, including local to global attention and ensembling and also prediction head, can actually be quite important for the performance of the model. So in conclusion, we designed a self-supervised learning approach for performing cell representation learning in the context of histopathology images without requiring any input annotations. This means now we have access to a massive amount of unlabeled data in histopathology. Next, we show that the learned representations in this framework can actually significantly and consistently outperform all of the existing baselines across multiple datasets and different tissue types. And also, as a last note, I have to mention that this model is not necessarily dependent on histopathology, so it can be applied on different datasets and also different applications. Please check out our paper for more details and more results. And thank you for your attention.